couldn't get here. So I've been called in at short notice to fill his shoes. And everyone knows that Lawrence has got some pretty big boots to fill, so if you can bear with me. My name is Peter Albertson and I work for Reef Catchments as a Sustainable Agricultural Coordinator. I am the Program Manager of the Reef Trust Partnership Mackay Whitsunday Water Quality Program, which is led by the Great Barrier Reef Foundation. This morning's session, we have 3 lots of 15-minute presentations this morning. They are HCPSL's Megan Zahmel, presenting their drone weed spraying trial. Number 2 is Becke Productivity Services, is Jasmine Connolly, presenting intercropping mixed species, and the third one is Canegrowers Innisfail Trap Services with Peter Becke and Charissa Rixon discussing subsurface mill ash mud. Following each presentation, there will be a few minutes for question time. To make it fair on everyone wanting to ask questions, if one question is taking too long to resolve, we will move on to another question and ask that initial question to be discussed outside of the session times. Also, basically on completion of the third presentation, questions, the presenters will move to the next room. Firstly, I'd like to call on our first presenter, Megan Zahmel. Thank you.
Okay, so good morning everyone. I hope you're enjoying your day. Today so my name's Megan Zahmel. I've been working for HCPSL since 2016 as the service provider for Project Catalyst. Today we are going to talk about the smarter weed control using drones trial that we've been operating at Michael Reinaudo's farm. So first of all, I'll just get Michael to introduce himself and tell us a little bit about his farming operations and why we wanted to do this trial.
Okay, hey, good morning everyone. So my name is Michael Reinaudo. We farm in the wet tropics. We are in Ingham. So we're a family farm where I work with my dad, my brother and my uncle, so there's the 4 of us. We've just recently, in the last couple of years, expanded our business, so our farms are pretty spread around the district. We've probably got maybe 70 kilometres between the 2 furthest farms that we have. So that's why when it comes to spraying, it's always that time of year, you're trying to get in there, you're trying to get it sprayed with the weather and you're trying to, bogging high rises and mucking around all the time. So we sort of thought to shift high rises from one farm to the other is always a big job because you've got to get pilots, you've got to make sure everyone's there and you've got to shift them around. So we had the drone. The reason we got the drone was basically to have a look at the bird's eye view of blocks and have a look what was happening in there. So I sort of said to Dad, look, before we shift the high rise, I'll slip out to that farm and I'll have a look and see if I can see any vines, if it's worth shifting out there. So we started doing that, and when we get those photos back, we sort of have a look and you could see what was happening. You could actually see vines, you could see different issues in the paddock. I said, look, it'd be great if you could actually just pinpoint those places where the vines were or the weeds were. So if you could actually pinpoint those places would be really helpful as well. So we started, doing that, and I talked to Megan and that about doing a trial, and we needed that expertise of Marcus to come in and just help us understand what sort of footage, what sort of photos we needed to be taking so he could mesh them all together and give us an actual photo of what was going on and then pinpoint. So yeah, that's why we sort of went down this path, and I'm sure we're going to be able to use this information or data for other things down the track as well. Thanks.
Okay, thanks Michael. So I'll just quickly touch on the trial outlooks. So first of all, we wanted to use high resolution imagery from the drones to map the weed infestations, and then we wanted to use these drones to apply herbicide precisely. That was available to us through Marcus Bolstrom and the DAF, so thank you to them. Our trial was aimed at targeting vine species in mature cane. The chemical recommended was MCPA and Starane. Unfortunately, and I'm not 100% sure why, the chemical that was actually used was Tordon at 1 litre per hectare. This has affected the results a little bit, but we've definitely still learnt a lot from the trial. So on this screen here we have the trial map from DAF and the drone view. We were able to, so Marcus went out there and he flew the drone over the block, picked up this imagery. He was then able to take this imagery with his program Drone Deploy and basically spit out this lovely little map to the far side there with the GPS points. They were then loaded up into the spray drone and the drone was able to go out and precisely target these positions.
It will descend to the height.
So
where
just a
the
couple of
radar
videos
lock on
of
the tank
the actual
is
footage.
in the mission of spraying. And let's have a look to see what it's doing. It's spraying, so it's, you can see the grid there that it's going at.
Oh, this is the grid you created?
Yep, so it's now, it's just about to be finished. And then once it's finished that, it should now come back to me here, which means it'll go up to, I think I've got to set it 20 metres and 30 metres. And then it'll come back and in theory, hopefully it'll land on the map. Doesn't really matter. It's not bad.
Marcus probably didn't realise he was going to be on video for everyone, so.
It operates completely different.
And then just another video. The drone itself actually holds 10 litres of water or chemical.
Better? Yeah, just needed that bit of height. There's the patch. Last year I had flowering cane. It was just, there's a maximum height that it'll spray above the cane. It won't go any further, it's got to lock on. And it was, second. Probably reduce that return home altitude, save that energy too. Alright, that's that one done. Downwind yet I can't smell it. The droplets that come out of this are huge. We'll put the element where it's especially so it's like rain coming out. Big wet droplets and very few fine. Once they send out with it, and we first put it on the water, there's a patch going up above it as there was below it.
So the drone is all GPS'd, which is why he was so confident to land it next to the water there. But there are a few little issues with the drone. It has a sensory that holds it 1 metre above the cane. You might not have heard very clearly in that video footage, but if you have mature flowering cane, that can affect that sensory. And other times, what we found also, if the vines had pulled the cane down or there was a bare patch, the drone wanted to go down into the cane canopy, but then couldn't quite figure out how to get itself out. Marcus was able to counteract this by putting the drone into manual operations and lifting it back up out of the cane. But there were just a few little issues that we come across straight away. So here are just some photos of before spraying, and then we went and flew the drone back over it 3 weeks after spraying, and you can sort of see that there are some signs of chemical uptake there with the browning off. Our main concern with this trial was actually missing weeds in the initial scout due to the full canopy of the mature cane, and when we went back into ground truth, we did actually find that that was an issue. So Daph and Marcus have done 3 years of trialling with the drones, and they have been successful in the past. Especially targeting red convolvulus vines, and he said that they have been quite successful on very thick patches of the convolvulus vine. We didn't get a great uptake of kill, and that's potentially because the recommended chemical wasn't used, the Tordon was used instead. Centro was the vine that we were targeting, and it can be quite hard to kill because of that area leaf. So we sort of believe that there's definitely more room and strongly recommended to do more trialling with chemicals and the water rates. Being that the drone can only take 10 litres of water, water rates is a big thing. And yes, once again, it was a little bit easy to miss those spots in the mature cane. So when we walked back through there from one spot to the other, we found areas that we just initially missed with the initial scout. In saying that though, I do believe that there is a lot more potential for the drone in smaller cane or younger cane. The photo to the far side is cane that was 4 months after harvest, and you can see the interrow a lot clearer, and therefore you can see the weeds a lot better. The cane that we were dealing with was the image closest to myself, and as you can see that the canopy is a lot more closed in and you can't quite see to the ground very well. So the results of the trial, and what did we learn? Unfortunately, we didn't quite get the results that we were hoping for, and that was due to several reasons. As I've said before, it was easy to miss the smaller infestation of weeds that were under that mature canopy or that thicker canopy. And then when we went and ground truthed it, we found that those weeds were sort of at a bit more of an out of hand stage. Their drone herbicide application, it did work on the smaller weeds, the blue top and things like that, but probably wasn't going to be enough to kill off the centro. As you can see there, we haven't had a lot of chemical uptake, but that potentially was due to the different chemicals, which we will do more trialling on, or recommended to do more trialling on. The drone definitely has more potential in the smaller cane where we can see better, and I think definitely weeds like sickle pod, GSP, and even guinea grass can definitely be mapped and perhaps targeted easier with the smaller cane and a clearer image of that interrow. And then we also believe that the drones themselves can be used just for mapping alone. So if you could go in there, have that map printed out to you, bring it to someone like Rod Nielsen, our GIS officer, he'd be able to put that into a prescription, you'd be able to put that into your tractor and go out and target those weeds in that precision agricultural manner, saving yourself and reducing your chemical usage. And then our conclusion. So, as I've said, I do believe that there's great potential for the drones in precision agriculture, even though we haven't had the greatest success. Future trialling should definitely be aimed at the chemicals and the low water rates, as I've mentioned. If we can get in there and get that earlier surveillance of weeds in the smaller cane crops, we're not having to deal with that really thick canopy cover, and mapping for time of effective application of land application via the tractors. So that is us. Thank you very much to Marcus Bolstrom and DAF for helping us out with the trial, and thank you very much to Reinaudo Farming Company for taking the time. As we all know, trials can be a little bit fiddly and time consuming, so thank you very much for that. And is there any questions? Yes, so you can, the drone goes at a certain level. Like I said, it will stay 1 metre above that cane, but then when you get the photos back, you can zoom in quite clearly and target and see the weeds more clearly. So yes. Yes, John.
I know it's going to be hard, but it'd be interesting to put it into context.
Look, I think what we're trying to do, from my perspective, the spraying with the drone is very limited. Like, we're still going to go there and have to spray with the high rise or put a plane in, because you're never going to get, because you know your farm with vines, it's never just a small patch. It's going to have to be done anyway. So to get to that point of what sort of water volumes we need to take out, I think it's probably a little bit of trial there, but for me it's about identifying where they are. I think that's what this has showed me. It's more about identifying where they are, and you know, whether you map them, give it to the high rise or whatever, it's going to be another form of spraying with the drone. Unless, you know, different weeds, whether it's guinea grass, sickle pod, whatever, arsenic weed, different sort of, you know, where you can actually mark them and go. But I think the water rate, you know, it's not going to be the, like, to save that trial.
Good question. Lawrence was supposed to be here to answer the chemical questions for me. Chemical's not exactly.
I mean, we can have that discussion. Is Roundup Roundup the go? Like, to get that water volume on that stool, you'd have to use something that's going to be taken in from the leaf, I assume. So.
Yeah, so we've got an option there potentially as well. Obviously, with guinea grass individual sections. But if you could perhaps target it in a smaller one, the cane's smaller and the guinea grass itself is smaller, yeah, there's definitely just potential there, but more trialling work needs to be done.
Any more questions? Can we just have you wait until the microphone gets to you, please?
Michael and Megan, do you intend carrying on with the trial? And if you do, will you then add one step further and go in and look at the financial aspect of that, as whether there will be how much financial, I suppose, gain for you by using that technology, so that you can target, I suppose, if you're going to target where your weeds are, what will be the financial impact?
So, yes, in some senses we will be continuing the trial on, but looking at more of a mapping tool than actually using the drone. Marcus's drone is one of, there's not enough of them basically. So the other thing is it only holds 10 litres, it does a lot of trips backwards and forwards sort of thing. So we sort of feel that there's a lot more potential to use this as a mapping tool. HCPSL then has all that equipment as well that we can offer that to our growers as a mapping tool by flying the drones over paddocks, creating the maps, and then being able to make a prescription for the tractor. So once we do that, then we can definitely look at economics, as I believe there would be a pretty big savings. You're definitely reducing your chemical if you can precisely target those things.
So I actually like the thought of the history as well, of like having known where the weeds were over a time frame, and just seeing it reduce as you go through as well.
Yes, Alan.
In this new technology, I know there's a lot of programs and photography work where they can distinguish different plants and feed it into a computer program. Do you think that has the potential to be loaded back into the high rise and control the high rise once you're in the paddock spraying, more so than using the drone to spray?
Definitely. It's like the prescription for fertiliser. You're going to switch it on, switch it off, where you're going, where you need it, where you don't need it. So that's definitely probably where you're headed, Alan, as opposed to spraying with the drone.
Yes, and I believe that's a lot more easier to uptake for a lot of growers as well. Most people have the GPS and the ability to put in the variable rate prescriptions. So definitely, that's the future of it.
And Jane, that would actually save a lot if you're doing just spraying where the vines are, as opposed to the whole paddock. Yeah, that would definitely be a financial benefit.
Okay, is there any more questions? Otherwise we better move on to the next speaker, or otherwise we'll run out of time. So I'd just like to thank our guest speaker first up, and if you could put your hands together, please. The next guest speaker is from the Becke Productivity Services, Jasmine, Connolly, and discussing intercropping mixed species. Jasmine.
Thanks, Peter. Good morning. I am Jasmine Connolly, and I am based in Becke Productivity Services. I've been there since July 2017 and been involved with Project Catalyst only recently. We've only come on board in the last year and a bit. So today we don't particularly have many conclusive results. So what I will be doing is just going through the broader adoption validation trials that we're involved with. Figure out how to work this thing. There we go. Okay, so we've got 5 broader adoption validation trials. And the first one that I will talk about is about irrigation efficiency. So we've actually got some that cover off on soil health, which seems to be quite a popular topic at these forums. Irrigation efficiencies and also alternative pest controls. So we'll kick off with SIB's irrigation efficiency trial. So he was interested to find if there was a way that he could improve his irrigation efficiencies by maximising his crops' water usage and managing his inflow rates. Prior to the commencement of this trial, he was unaware of the amount of water he was applying to the crop and the crop's requirement and demand. To start the trial off, we decided that we would create a readily available water map, which is on the far left. This shows the water that's available to the cane And you can see it varies. I don't know if you can see that very well, but the different colours indicate different millimetres available. So this is based off of his soil map, and pretty much the next step was to install a moisture meter. So we've got a G dot there, you can see, which is the next picture, and we calibrated this to the growth of the cane so we could figure out when the trigger point to irrigate was. This was around 2 dots, I believe it was. Then what we did was we assessed his pumps. So we had an ultrasonic flow meter, and we strapped this onto the delivery pipe of his pumps, and we could figure out how many litres a second he was putting on by doing this. So then from there we worked out how many hours he was irrigating that block and what the area was, and we could figure out how many megalitres per hectare he was applying. Now that we had all of this information, we could put this into an irrigation scheduling program called Irrigweb, which is on the right-hand side. Irrigweb is a very useful tool, and it can help monitor and schedule irrigations. It takes into account weather from BOM, so it can take into account rainfall, how many sunny or cloudy days there are. It also takes into account the soil type and water holding capacity, and it essentially grows a cane crop in the program using the information you put in. So this can be very useful, and if the correct information is applied, it can be very accurate as well. So once we had Irrigweb up and going, we could put the information in, and pretty much, let's see if this works. Yep. So you've got these blue bars, which are your irrigations. Your green bar is your rainfall events, so that's from BOM. You've got an orange line here that is your deficit or your trigger point to irrigate, and the red line is pretty much deficit that bounces around. As you'll see, when you irrigate, it comes straight back up. As it starts to go down closer to that point, it's the trigger to irrigate. So this is something that you can monitor from your computer or your phone. So now that we had all of that information in there, we could ground truth it as well with the G-dot. So yeah, pretty much a G-dot was really good because it's real-time data and it's in field, so we could ground truth Irrigweb and make sure that everything was working correctly. So Sib can now just drive around his farm with confidence and have a look at his G-dot and know that it needs irrigating at a certain point, like the 2 dots as I said before. He now enters his irrigations into Irrigweb, and as the season goes on, we'll be able to see how much he has used, how many megs per hectare, and look at his cane yield, and maybe see if there's any areas that we can hopefully pick up in irrigation, whether it's to irrigate more or irrigate less. We'll look into that a bit further and see how his cane yield responds to that as well. Alright, so we'll move on to the soil health trials at the moment. So we've got a few there. The first one was at Rich's farm. So his previous practice was to plant cane on cane and have a few months fallow in between. And he'd never used a legume crop as a cover crop and was interested to see if it could work on his farm for soil health benefits, so he didn't actually harvest this crop. He wanted a legume that had minimal input costs, so he decided that ebony cowpea would be the best fit. The crop was brought up on moisture from rain in February of 2020 and grown for only 2 months. So just before the flowers started to form, that's pretty much how far we grew it to. And we only grew the cowpea for a short period due to him wanting to plant cane in April, and also to avoid the production of seed pods that could potentially, if they dropped into the soil, grow up in the cane and impact his cane growth. We dug up some of the cowpea just before it was sprayed out to have a look at the root structure and the nodules, which you can see here. We were quite surprised at the size they'd grown to in only a couple of months. Once it was sprayed out, it was left to desiccate before being mulched into the soil, and cane was planted shortly after this. He also voluntarily reduced his nitrogen by 70 kilos a hectare as per the 6 easy steps. He did not want to potentially sacrifice tonnes for CCS. And Richard is so far impressed with how his block looks, and he's doing this to all of his fallow blocks now, which is a good result, I guess. Gary is another grower who was interested in improving his soil health. Fortunately, Gary had some mixed seed that contained corn, cowpea, tillage radish, lab, and mung bean, and we planted this in May 2020 with a seed spreader, and the seeds were irrigated in, so there was no cultivation. Gary also had the idea to graze his cattle in the block once the mixed species had come up, to provide them with a food source and to also give the soil some additional nutritional benefits. And also Gary wanted minimal input costs, so irrigation was not frequent. However, when it was, the EC of the water was relatively high, just due to the nature of where he's located. And this didn't particularly benefit the mixed species either. So we did a suite of microbial tests, which were taken from 4 points across the block to assess fungal, bacterial, and nematode presence. The first test was done before the mixed species was planted, over here, to create a baseline in the weedy fallow. And the next test was done when the mixed species was actively growing. So originally the cattle was actually meant to be put in shortly after this period. However, unforeseen circumstances, they've only just actually gotten in the block now. So that's a photo from last week. So once the cattle are moved on, we'll probably take some more tests, and we'll just have a look at how the cattle have impacted the soil. Due to the salt issue identified, a crop of sorghum may be planted to actually help remove salts, and the cattle will also graze this as well. And yeah, pretty much once we have all the biological samples, we will compare the progression. Mark was another grower who was interested in improving his soil health, and he wanted to do this through soil amelioration and fallow cropping. He was already involved with the trial with BPS and Sugar Research Australia, which was looking at using green waste from Townsville and Burdekin City Councils, and mill mud also from Invicta Mill. The trial commenced in December 2019, where 3 treatments were applied to the fallow block. These were green waste at 96 tonnes a hectare, mill mud at 80 tonnes a hectare, and a mixture of 50-50 mill mud to green waste at 35 tonnes a hectare, and there was a control, of course. The products were applied using a BC cartage truck with a walking floor and a spreader. A6785 soybeans were planted a few weeks later across the whole trial block and harvested for grain in April 2020. Cane was then planted in May of 2020 as well. So the same types of biological testing were done on this trial as well. A sample was taken as a baseline at the beginning, and another when the soybeans were growing, and then another when the cane was planted to monitor fungal and bacterial levels. Worm counts were also conducted, and it was found that the control had half of the worms of all the other treatments. So I've got a few graphs up there. I'll just quickly sort of explain the basis of that. So from the biological test regarding the fungal biomass results, in the first graph over here, we can see that the control, which is that blue bar, has stayed relatively constant from the legumes to planting cane. So this indicates that the treatments are having an effect. Some of the treatments decreased after the initial baseline test. This could be due to the tillage that was required to establish the trial, which will obviously disturb the fungal community. This was a huge there was a huge boost in green waste. However, it was only short-lived. You can see it's dropped down there. But overall, the fungal biomass increased from the combination of green waste and mill mud, and also the mill mud by itself. If we look at the next graph here, which is the estimate of total microbial activity, we see the original baseline test is much lower than all the other treatments. And there has been a steady increase in the green waste mill mud mix and the mill mud by itself, which is similar to what we saw in the previous graph over here. So the last graph down here is the labile carbon between treatments. So the original baseline test was quite high, and then most treatments tend to drop off. Again, this may reflect the tillage that's required to establish the trial. The first sample taken in the green waste was the highest, and this may come down to the huge concentration of labile carbon that's actually in the green waste compared to the other treatments. This was short-lived, however, and reflect the spike that we saw in the other 2 graphs. It then decreased in the second sample when cane was planted. So these are not the final results, and there is much more data to be interpreted. But I guess what we can take away from these biological analyses is that we want to increase the soil diversity, and particularly the fungal community, due to their soil structure and nutrient cycling abilities. Higher the amount of carbon in the soil means greater diversity of life, even if it is short-lived. This could play crucial roles, particularly when establishing or germinating a crop. However, biological systems can be hard to monitor and make sense of because there are variables that influence the environment, such as soil temperature and moisture. Even management practices can have an influence, particularly tillage and management of organic inputs. And understanding the best time to take samples is something that we may need to work more on. John has historically had issues with root knot and lesion nematode and wanted to find if there was an alternative way of controlling them in his sugarcane crop. Now, he anecdotally heard stories of veggie growers using molasses to control for nematodes in the past and thought that maybe this could work on his cane crop if he could find a way to apply it. Luckily, John came across an implement from another region and modified to suit the trial as a subsurface liquid side dresser. The idea behind applying molasses was to increase the biodiversity of the soil biome by providing a food source so that parasitic nematodes were not the predominant group. By doing this, beneficial microbes, fungi, and nematodes would respond to the molasses and create a more complex food web that could help suppress parasitic nematodes. We obtained molasses from the local stock feeds and calibrated the applicator to have a 1-to-1 ratio of water to molasses, and this was mixed into an 800-litre tank. There were 2 treatments of molasses mixture. One was applied at 500 litres a hectare, and the second treatment was also 500 litres a hectare. However, this was applied with 4 weeks in between. Nematode samples were taken before application across the block to create a baseline, and then the second lot were taken a few weeks after application. The level of beneficials from the baseline reading was already higher than expected, which is good for John. There does not seem to be anything outstanding according to the nematode counts, except that maybe 2 applications could be better than 1. As you can see, there's quite a difference there. So it could have potentially suppressed the the parasitic by having 2 applications, whereas 1 application sort of boosted everything. So the trial will be taken to harvest and each plot will be analysed separately, but we may see something in the harvest data. Now, the last broader adoption trial is at David and Marty's, so you would have seen the video already this morning. David had been trialling the use of legumes on his cane farm for the last few years, and he's seen soil health benefits and economic return. This time, he was interested in intercropping coriander soybean and tillage radish into his existing sugarcane return crop to improve soil health. So the idea behind this was that the soybean would provide nitrogen and the tillage radish would pull nutrients from deeper in the soil profile and bust through compaction with its taproot. Once the cane canopy closes in, the radish and soy will die off and the nutrients will become available to the cane. So we had 3 treatments. There was soybean, tillage radish, and a mixture of the 2. So we applied the seed using a row crop planter, which buried the seeds directly either side of the cane stool on the shoulder of the row in October 2020. And because the Burdekin is fully irrigated, germination and growth of the interrow crops should not be compromised due to lack of soil, moisture, and competition with the cane. There is also an opportunity to reduce the amount of chemical used to control weeds due to the shade-out effect that the interrow crops have. The benefits for the soil, having 3 different types of plants, a legume, a grass, and a brassica, will promote the growth of different fungi, bacteria, and other microbes in the soil biome. The return cane N fertiliser was reduced by 80 kilos a hectare to see if the interrow crops could actually match the cane next to it outside of the trial, which had his normal rate of fertiliser. What we've seen in the trial so far is that the tillage radish has not grown as large as we had hoped. However, what we are finding is that a lot of the taproots tend to have a bend, sort of like this one, sort of like a dog leg, rather than the nice straight roots that you see like at Razor Moras. We believe this could be due to a compaction layer that the root is trying to get through. The soybeans are acting as we thought that they would. They tend to be quite elongated and straggly due to competition for light, but the trial is not yet finished. So the canopy of the cane is starting to close in now, and what we'll see is that the soys and radish will start to die off, and we may see the benefits to the cane with the interrow crops pretty much producing the nutrients available. One thing we may look at, since we've seen this happening in a lot of the tillage radish, is using a penetrometer to measure the amount of compaction. And when the crushing starts, we'll harvest the treatments individually and see if there is any difference in the tonnes, CCS and tonnes of sugar per hectare. So I'd just like to make a few acknowledgements. Obviously the growers involved in Project Catalyst is greatly appreciated. Sugar Research Australia for all their collaborative work and assistance, and the UQ Uni for analysing the biological testing.
Thank you very much, Jasmine. If we've got a couple of questions now, we've only got a few minutes before we have to get on to the next speaker. So could you just put your hand up and we'll get the microphone to you.
That companion planting, it's been tossed around for a long time. How come it's never become mainstream? You know, it seems to be a good fit. Johnny's done it, and I don't know why.
Yeah, so I guess that's the reason why we're doing it. We're looking into if it's beneficial for the cane. Yeah, I can't answer that. I'm relatively new to the industry, so yeah.
About 10 years ago, we did companion planting with soybeans and mung beans. It wasn't irrigated, and we found out that they were competing for moisture later on in the year. And also we had an influx of rats that actually, out of one of the years, ate the whole trial out.
Yeah, that's not great. Okay, well that's interesting, something to look for. But yeah, we're hoping with the irrigation, there won't be that competition for moisture. Yep.
Okay, if there's no other questions, I'd like you to thank Jasmine for her efforts. And I'd like to welcome Canegrowers Innisfail Trap Services, Peter Becke and Charissa Rixon. They'll be discussing subsurface mill ash mud. Thank you.
Oh, g'day. I'm Peter Becke from Canegrowers Innisfail. I've been the extension provider up there for 3 years now, and we got Joe Morano's subsurface ash trial that we'll be presenting today alongside of Charissa from Trap and her trial with subsurface mud. Anyway, really hate doing these things. Yeah, you got it. All right, start off, I'll get Joe to give a bit of background into the reasons that he was interested in having a look at going to subsurface ash and the background of his farm.
Yeah, thanks Peter and Charissa. We farm in the new harbour line area of Marillion. It's a very sandy soil, commonly known as Marillion sands. We can grow good crops in plant cane, but they drop off very quickly. We don't lose still, but the cane just doesn't perform like it normally does, like in a plant crop. So we've been trialing mud and ash over a number of years. We've tried biochar, we've tried bentonite, we've tried multiple things, we've tried irrigation. None of them, well, we probably used them at too low a rates, and I'm a firm believer you start at a high rate and you work down, you don't start at a low rate and work up. But the ash we've tried broadcasting, we've tried on top of the stool, and currently we're trying to get it under the stool, but at the moment I don't want to go out and spend a lot of money, because if it's no different than just applying on top, you spend all that money for no reason. So getting it under the ground and below the set is the biggest challenge, especially in sandy country, because it doesn't, if you lay it on top and then you try and pull it in with a ripper, it doesn't fall in because the sand caves in. I know people have trenched it in and stuff like that, but our trenches wouldn't hold. So yeah, that's why we want to maintain production, a higher production in return crops rather than just replanting or leaving fallow and planting. Commonly, You know, in return cane, we probably only average about 70 tonne per hectare. In plant crops, like I said, we can get up to 120, 130 tonne per hectare, but it drops off really quickly. So I think back in 2014, when we had the GIVE conference in Innisfail, we talked about this, and people were trying at a low rate. And the problem is, if we go through and furrow before the spreader comes through, we don't have hills, but we have sidelings, and a trailing ash spreader falls in, so you don't actually get the ash into the furrow. So there's multiple challenges, and that's why I got involved with it.
So for the trial, basically we set it up as a replicated complete block trial. We've basically gone through with 3 replicates running subsurface ash, no ash, and just surface applied ash, just to have a look at obviously whether we're actually getting the benefit from just the ash or where the benefit comes into your subsurface application as well. So the implementation went through with Joe opening up a furrow with a modified bed renovator, just taking the covering discs off to pull that out. Then once that was done, we dropped the ash into the furrow using the spreader. As Joe was just saying before, with that sand sort of soil that didn't really track too true on top, and we had quite a lot of issues with trying to get that in. Then the ash was covered back in with the bed renovator with the covering discs on, and again, as you can sort of see in that photo there's a little bit of spots where you can still see the ash on the surface, and then we just planted straight into that.
So at George Henry's we did something quite similar. He looked at a mill mud and ash, so he was looking at rates of between 25 and 50 tonnes of mill mud plus the 25 to 50 tonnes of ash as well. He started it because he's got cane that the roots aren't going down and not performing. Originally thought it was a drainage issue, a subsurface drainage, because it was quite a wet area on his farm. He's implemented subsurface drainage and he's found out the roots are now hitting a zone and just burning off, and they're not going any deeper. So he's looking at it as a soil health type issue to see if he can help attract his roots further deeper into the soil. So he put it in with he uses a disc opener to open it up. And this is a machine that George built himself. So he actually puts 3 rows out at a time of mill mud and ash. So he gets the mill mud and ash delivered on farm, and then he uses this pre-planting.
So what we've found for the plant and first return crop that we've harvested and watched was that while there wasn't any statistical significance of the ash treatments in yield or CCS, there are a few spots where things stood out, and we're definitely interested in trying to have another go at it where we can get it done properly and banded down deeper.
So with George's trials, we only got to harvest the plant cane because 2020 decided to be difficult. The harvest round where he was due to harvest, well, when he got the first harvest round, it was actually wet and tully, and then didn't rain after that. And that was because it's had subsurface drainage applied, it was the driest block. So after getting bogged elsewhere, they went and took the trial site off, and they didn't even notify me, so we didn't get the results. Wasn't a fully replicated trial because we didn't have the area and that to do it. But George they're the rates he's used, and there is a general trend that as your mud and ash rate went up, his yields went up. Now, he didn't change his fertiliser at all with this. The other thing that we've done, because we couldn't do the yields, we looked at chemical soil testing and biological soil testing. I don't have the biology results back yet, but we did depths at 0 to 20 and 40 to 60. Some very interesting things to note. Organic carbon. 0 to 20, it's up around about the 0.91 mark except for one. When we get to the 40 to 60, we're down about 0.4. So and this is taken in, so the 0 to 20 and the 40 to 60 are taken out of the exact same hole, so in the exact same sites and bundled up. So his topsoil has got reasonable organic carbon, but his subsurface has got basically bugger all. The other thing to note is what we found is with his calcium, but the lime treatment on its own, the mud and ash at 25 tonne to the hectare was about the same. By doing 50 tonne to the hectare, it was a lot better. And that carried through a lot more into the subsurface. So the lime didn't seem to raise the subsurface much, but the mud ash mixture did. The other thing that really interested George, because he was only putting the 25 or the 50 tonne to the hectare on, he hadn't cut his phosphorus rates yet because it's not mandated to do that. But what really surprised him is how high those rates when banded zonally under the stool, and we took this on the shoulder of the stool, not dead centre, how much that raised his phosphorus levels in the soil compared to, you can look at your lime as your standard, and it even raised it in the double rate at the subsurface. The other thing that raised was his potassium.
Basically, in summary, the main things that we had the issues with getting the band in, and as Joe was saying, he really wants to get that higher rate in there just to assess it properly. We're also, as Joe was saying, wanting to continue to see the benefit in the older ratoons, so we'll be keeping an eye on that as it gets older, just to see if there's something that starts to come out of it as that's gone through. And the main one is that the subsurface application is a time-consuming sort of a process, and we want to be sure that it's the right way to go before throwing big dollars into equipment.
And just for George's trial, basically there he's just seeing that yes, it is helping him in his situation. He's quite surprised at the improvement in the chemical properties. We're still, as I said, we're still waiting for the biological. And for him, he's a long way from the mill, so it makes the application of mud and ash more affordable and can go over large areas. Any more questions? Talk to Peter.
Okay, thank you very much for that. If everyone put their hands together. Now we've got a few minutes up our sleeve for some more questions, so could we start, please?
Yeah, we subsurface mill mud and ash at 50 tonnes per hectare, two separate jobs. But we, to get the ash down deeper, the depth we do is, you could easily say, 2 beer cans deep. That's how, but we want to go deeper. We put run through with a single tine ripper each row, and the void, we hope it will send the ash and the filter press down to whatever depth you want. Well, that's what we do to get it down any deeper. You can't physically go any deeper with. We use Gesner furrow boards, so we can't physically go any deeper with it. So we use a ripper leg, hopefully, to make the void to go send the material down.
Any more questions? Okay. I'll have one question of my own. Have you had any communications with Wilmar, with their mill mud applications and things that trials they've been doing? They've shown that some of these trials they've had for quite a few years, that it's certainly a long-term benefit that you're getting out of it, not just a short-term. So are you expecting to take these trials on for a long period of time as well?
Just from George's perspective, even though we've only done this for 2 years, he's quite happy he's quite satisfied with where he's at, and he's convinced that it's going to benefit his system anyway. Yeah, so I don't think he's going to be pushing to take it on any further. He's built all his own equipment for putting the mud down and for opening up the drill. He's just bought another piece of equipment to cut up to make a 3-row machine for V'ing out, for making these things. So quite ingenious at doing all that side of it. But he's just after that little initial, qualification and quantification, and now we've got that, he's going, yeah, I think I'll benefit from it anyway, so he's happy.
I'll keep using ash. No, I'll keep using ash. I'm just trying to perfect the way. I mean, I do know there's a benefit in ash, and in a block we applied ash 5 years ago. It's fallow now. We don't have to add, and in our sandy soils, it's taboo not to apply calcium before you plant, but the soil test results come back saying we don't need to apply any calcium, which I'll probably still apply some because it's in my DNA. Yeah, so we'll keep using ash. I want to try and get longevity out of it. So if I can get it below and it makes a difference, I'll keep trying to get that, and before I knock it and just keep applying it on top.
And just one, from my perspective, Plain Creek always had issues with distribution of mill mud and ash, etc. Do you have any issues like that where you are?
As I said, I'm fortunate I'm in sandy soil, so when it rains, no one else wants it, so they bring it to me.
Okay, is there any other questions while we're here? Okay, thank you very much. If you could put your hands...
Yeah, under the new regulations, if you're not allowed to apply, I think if you're not allowed to apply phosphorus according to your soil test, I don't think you're supposed to put mill mud or ash on. So it's just going to come back down to doing individual soil tests each year on each block.
Thank you very much for your questions. It is time for our presenters to change rooms now, as Jason has finished in the next room. Thank you very much. Give them a big round of applause for those trials, and you'll be able to talk to them during lunch.